Schindler, about a quantitative examination for the extent of cohort dominance in sympatric Alaskan salmonids. So the core of my study rests with a challenge that conservationists, managers, and ecologists currently face, is that while we can look at a population and describe the population dynamics, we don't know all the specific drivers that cause them. So for example, we can look at a population and assign a general trend and say it's a stable or a stochastic or a cyclic population dynamic that changes over time. And we'd like to know that to inform our environmental decisions, but we don't understand all the drivers of them. So just to give you an idea of what I'm talking about here, here is kind of an idealized model of two different population dynamics we might see in a wild organism. So in the black here is what we consider a stable population growth rate, where the population over time, it grows at a relatively steady rate and levels out and maxes at a certain carrying capacity that the ecosystem can handle. However, for my study, I'm investigating a possible cyclic population dynamic here. So there's a number of different ways you can interpret what exactly that means, what exactly are we seeing when we say cyclic population dynamic. But in this instance and in this model, I'm looking at periods where population size spikes and then has a long period of a low growth, if not decline, before spiking again. And the important part is that these rapid increases in population occur at consistent time intervals. One possible cause of these cyclic population dynamics is something called the storage effect, where species with stochastic recruitment patterns often don't always recruit, but when they do recruit, what happens is that the cohort that recruits becomes dominant, and that through things like intracivic competition, cannibalism, they outcompete subsequent cohorts and become the most prevalent in the population. And these often are reflected in the age and size dynamics of these organisms. So again, to kind of give you a visual, on the top is what we could say is a stable age-size distribution, where as time goes on, on the x-axis is our age and size groups, and our y-axis is our population size. In a stable population distribution, over time we'd see relatively the same number of, say, juveniles or mature adults and spawners. That relationship, that ratio is largely the same over time. However, under storage effect, what happens is that we start with after, say, who knows how many years of failed recruitments, we have a successful recruitment event of juveniles. But because of storage effect and cohort dominance, this cohort is suppressing the subsequent recruitment of new juveniles next year. So what we see as time goes on is one cohort is overwhelmingly representative of the population, and so as size and age increases over time. And that brings me to my species that I study, which are actually 3 species of Alaskan salmonids. And I study them because they are known to have a number of factors that might lead to storage effects, such as consistent annual spawning patterns, highly variable juvenile survivorship, and strong competition for resources, especially in their freshwater environments. And so I evaluated 3 different species for this phenomena, which are Arctic char, Arctic grayling, and rainbow trout. And the objective of this study is to determine if these populations of previously unstudied Alaskan Arctic char, Arctic grayling, and rainbow trout are experiencing cyclic population dynamics. And also secondly, if they are experiencing cyclic population dynamics, are these patterns driven by the dominant cohorts brought on by storage effect, or is it something else? And so my advisor and I, we came up with kind of 2 hypotheses for our different species. For Arctic char, we believe that the population size will experience periods of about 10 to 15 years where that strong cohort has recruited into the population and is remaining dominant and suppressing the recruitment of subsequent generations. For about 10 to 15 years, we assigned that because that is the natural lifespan, approximately, of Arctic char. For Arctic grayling and rainbow trout, we hypothesized something similar, where it will experience cohort dominance through cyclic population dynamics, but at shorter intervals compared to Arctic char, simply because they have shorter lifespans. So they recruit to the population, live for an X number of years, dominate and suppress the subsequent recruitment of new cohorts, and then die, and this process repeats. So our study area takes us to the Wood River system in southwestern Alaska between 3 tributaries. In the top left here, you can see this is Little Togiak River, where from 1972 to 2023, Arctic char were sampled every year. And on the right here, on these two bodies of water, this is Lynx Creek and Hidden Creek. This is where rainbow trout and Arctic grayling were sampled from 2000 to 2023. And during these samples, size was taken, sizes of each fish sample was taken and used in the analysis for cohort dominance. How exactly did I do that? Well, I used something called a wavelet analysis for periodicity. So basically, with the body sizes collected under fork length from each body of water, I averaged body size per year and ran them through this analysis in R, which basically generates certain wavelet power and significance for periodicity. And so periodicity, in simplest terms, is how many years it takes for that average body size to oscillate from a low back up to a high and low to again. And so that signifies, we use that as a proxy to signify cyclic population dynamics, where you'd expect under a cyclic population, if these populations are experiencing such dynamic, we expect to see a low followed by a rise, possibly a dominance if storage effect is occurring, and then a subsequent fall, and then repeated iterations of that over our time series in a cyclic pattern. As for the wavelet model itself, our goal is to determine one, does this model generate high wavelet power denoting that periodicity like I just mentioned, and two, when it comes to storage effects, if these periods of high power and significance correspond directly with the natural lifespans of each species, does that indicate cohort dominance and storage effects? So for Arctic char, which was originally the focus of my study, on the left here is our kind of a wavelet power distribution produced by the model. So our x-axis is our time series from 1972 to 2023. Our y-axis is our periodicity. So basically, how many years does it take for the average body size in the population to start at a low, rise up, and then fall again? And in the red, the color to pay attention to is kind of the red here, as denoting what periodicities have the highest wavelet power, and that's aggregated in this right figure here, where basically wavelet power for each periodicity is averaged and deemed significant. So as you can see here, from about roughly, I'd say that's maybe 15 to 11 or so years periodicity for, like I said, for the average body size of Arctic char and Little Togig River to, at a low, to increase, and then subsequently decrease at a periodic, at a consistent interval over our time series. Similarly for Arctic grayling and rainbow trout, Arctic grayling had, the model had a difficult time deeming any significance for either species in Hidden Lake. However, in Lynx Creek, both species we can see from about 10 to 14 years, there's significant periodicity for Arctic grayling, and in Lynx Creek, for about 7 to 14 years, there's significant periodicity for rainbow trout. And kind of summary results here between our 5 species. The important one to take away here is that our range of significant periods and the periodicity that the model basically selected for the most power is roughly the same as the natural lifespan of Arctic char. So what that means is that the periodicity, when we're seeing those oscillations, is directly proportional, is directly aligning, not necessarily proportional, but directly aligns with the natural generation time of Arctic char. So yeah, to kind of elaborate on that a little bit, for Arctic char and Little Togig River, we can determine that they are experiencing cyclic population dynamics because as indicated in the red, there is strong periodicity present. And because that periodicity is corresponding with high natural lifespans, what that means is that when we see an oscillation in average body size, that is reflective of the recruitment, growth, dominance, and then death of that cohort. For Arctic grayling and rainbow trout in the Wood River system, they are experiencing cyclic patterns of population because, again, we did denote that the wavelet model does believe that there is periodicity in the average body size, but since they did not correspond with their natural lifespans, we are uncertain if storage effect is the case. I'd like to thank my advisor for his guidance and his support for this project. I'd also like to thank the Alaskan Salmon Program for helping extract the data.
So is it possible cycles that you do in an environmental effect, like for example, the size of the sockeye salmon now?
Yeah, so that's actually something I looked into the literature relatively recently. And so you're correct in that all three species are dependent on the sockeye salmon within this region. So one thing I read in literature is that the cannibalistic behavior of Arctic char in particular is heightened when there's no other fish species present. So I believe that if, say, the sockeye salmon run is poor, there might be higher rates of cannibalism among Arctic char and thus facilitate that dominant cohort in the population. That's one possibility worth investigating. Thank you.